We're gonna be doing this over 2 days. We've already kind of, 3 days, I guess, but we already started talking about it last class and having you start with your objectives and hypotheses. Today, I'm gonna give you a little more background and make sure everyone's on the same page. But then we're also gonna continue this lab on Thursday and talk more about the detailed analysis of it. I want to make sure I don't spend too much time talking that we run out of time to actually get our data. So, but I do want to make sure that we have the basic grounding of what are we doing, why are we doing it, what are we looking for. Okay. What is the objective of our lab today, this enzyme lab?
What is the relationship of substrate concentration?
Yeah, that's exactly it. It's, you know, let's say what is the substrate concentration on reaction rate? Or you could say, like, you know, how is reaction rate affected by substrate concentration, or however you want to do it. I'll reiterate because I got a bunch of objectives that were kind of a little more mixed up. Your objective should always be just a simple question. It has to be something, and it should directly have, in this case, we have our independent variable, which is the substrate concentration, our dependent variable, which is the reaction rate. It should be really simple and clear. You know, that is the first step in actually successfully doing any kind of science, is just being really focused on what exactly are we trying to accomplish and answer here. So the objective is usually really straightforward and simple.
So the objective should say like what the substrate is and what.
No, because we don't actually care what we're gonna, when we design the actual experiment, we like to come up with an actual reaction to study. But mainly we're looking at just any enzymatically catalyzed reaction. Yeah, so we should say, what is the effect of substrate concentration on reaction rate? We could say, like, you know, an enzyme reaction. If we wanted to be a little more explicit, you know, in an enzymatically catalyzed reaction, when the enzyme is held constant. So you could say that. If you just wrote what I wrote in black and the rest was implied, I wouldn't actually be fine with that either. So again, our independent variable is substrate concentration. You guys should write this down. Brackets concentration. Yeah, what was actually was clear to me from some questions earlier today is people I think are still like not exactly comfortable with the idea of concentration. I think concentration is just like a ratio. You know, you have some solution and it's like a solute. Solute is something dissolved in it. The solvent is usually water in our case. Solute is stuff in here. The concentration is, you know, higher concentration is more solute for a certain amount of solvent or solution. You know, less concentration. And so in our experiment, we're doing hydrogen peroxide as our substrate, right? Our enzymatically catalyzed reaction that we're going to use as our test for today is going to just be hydrogen peroxide, H2O2, which is a liquid. It looks basically, you've seen it, it looks like water. It's way more reactive. You know, it breaks apart into water, which is also obvious, and oxygen. And typically you write 1/2 O2, because the reality is it's going to be oxygen molecule when we come off of this thing. So this is a gas. So this is the reaction that we're going to be looking at. This is going to be, and this will maybe happen really slowly on its own, but we want to make it go fast. We use an enzyme. So we're going to have an enzyme called catalase, which will catalyze this to make it go much faster. This is something that is made in your liver. We're going to actually you want to Woodland Market. We have fresh calf liver. This is where we're going to get our enzymes. We're actually going to take our calf liver and we are going to put it in the blender and make a liver smoothie here. And that is how we're going to actually get our enzymes. Just going to use H2O2 is very reactive, so it's your body when it shows up. So this is going to be our reaction we're going to look at. Part of the reason this is, so let's start with the substrate concentration or independent variable. To change different values of our independent variable, how are we going to do that? How can we just in a practical way plug in different substrate concentrations, given that this is just an actual thing. In fact, hydrogen peroxide is 3% hydrogen peroxide. Hydrogen peroxide is going to but how am I going to, adjust to different substrate concentrations?
Add distilled water to it.
We can, in fact, we're gonna always have like 5 mils. One of them we could have. I have just 5 mils of distilled water. What's the concentration of hydrogen peroxide in there? 0. I have 5 mils, and it's just pure, I'm going to say H2O2, but in reality, it's the 3% H2O2. This would be like 100%. And then I can just do, a series of dilutions in between, which we're going to do. We're going to do 1 part H2O2 and 4 parts water. And then it'll be 2 parts water, 2 parts H2O2, 3 parts water. And then it'll be 3 parts H2O2 and 2 parts water. And then 4 parts H2O2 and 1 part water. And then this is just pure hydrogen peroxide. So we'll have 5 different dilutions, all the way from 0 to 100%. So that's going to take care of our independent variable. We'll just create these different dilutions from 0 concentration to 100% concentration of hydrogen peroxide solutions that we're going to put into our reactor. Does that make sense? How we're going to, so measuring Measuring the independent variable is easy. We just mix up the proper proportion of water to hydrogen peroxide, and we got our for that particular trial. This part is where it's going to get more complicated. How are we going to find some way to actually kind of quantify and measure the reaction rate? So we're going to do exactly. So one of the nice things, the oxygen is a gas. Right? When H2O2, this is hydrogen peroxide, becomes H2O, we don't see anything happening in terms of it. Water and hydrogen peroxide look the same. But oxygen gas are these little bubbles that are coming out. Not only that, a mole of water is just like a little pedal in your hand. What is a mole of an ideal gas? What's the volume of a mole of an ideal gas?
Liter. Never mind.
No, it's quite a bit more actually. Actually, cubic liter wouldn't make sense because a liter is a volume. People remember
It's
this?
like a large amount because the molecules are just.
Yeah, so it's I think it's like 22.4 liters, like at standard temperature and pressure. Right, so this is going to be really noticeable. The volume of the gas or the bubbles being produced are going to be noticeable. We'll see the bubbles. We can catch them. We're going to catch them. That's what we got these burettes for. We're going to have these burettes initially filled with water, but as the reaction goes, we have the little reaction vessel out through a little hose. The bubbles are going to come up here, and we can actually see the volume of oxygen being produced as it displaces the water in the burette. So the first step is we'll pick a particular substrate concentration that we're going to do for some trial. And we talked about enzyme held constant. We're going to have our set amount of enzyme in the picture. And then we're going to get this thing to run. And we're going to see the bubbles, and we're going to notice the bubbles being produced. And that's just the first step, though. That's still not a reaction rate. So what I'm going to tell you really briefly, and then we're going to talk about in more detail, is how the heck do we go from seeing bubbles coming out to actually coming up with an actual number that we're going to use as a reaction rate for that particular condition. So you're going to have a thing like time, the mils of oxygen. And you're going to do it every 5 seconds. And every 5 seconds, you're going to see how much oxygen have we made now? How much have we made now? How much have we made now? So this is going to start going up. Right? The more this reaction goes, the more of the H2O2 breaking down and becoming oxygen, the more oxygen is going to be produced. So we're going to see some graph like this. So this is like for one trial. Time, seconds, see mils of oxygen produced. And what are you going to expect?
It's gonna go up.
Right, the longer it goes, the more oxygen gets made, and we see the total amount of oxygen produced is going up. Eventually, what's going to happen?
It's gonna plateau.
Plateau. Why? What do you mean? Why is that?
The enzymes will all be engaged.
No, actually.
The enzymes will run out of places to bind to.
This is where people get confused here. So saturation, you can't see saturation from looking at one trial. If you're looking at one trial, there's no way you can know if enzymes are being saturated or not. You're just going to see what the rate is for that trial. The only way to know if you have saturation is if we're increasing the concentration on another trial, but the reaction rate didn't get faster. So you can only see saturation of the enzyme if you're looking at multiple trials with different enzyme companies, different substrate concentrations. John?
I was just going to answer your original question. Plateau as a reaction runs to completion.
Exactly. So eventually, all the hydrogen peroxide that you started with is broken down. There's nothing else to break down. There could be enzymes floating around, but there's no substrate. So eventually, reaction is over. You know, all H2O has broken down. So this is really important to kind of get straight, because the picture, the graphs we're going to draw that look like it's saturation, they're going to superficially look similar to this, but something super different going on. The axes are going to be different, but people get really confused because the two things look similar superficially in terms of the shape. You know, it'd be like, you know, a bowling ball and an old bomb from a Bugs Bunny cartoon look almost the same, but they're really different things. You don't want to confuse them because you're going to get into trouble. It's the same thing. If you're looking at this graph that looks like it's rising up and flattens out, it's going to look similar when we look at another graph later on that actually is describing enzyme saturation, but this one has nothing to do directly with showing any kind of saturation. This is just showing the reaction done. The reaction is over, keeping out, because all the substrates broken down. So what we're gonna do is we're gonna run a reaction. Let's say this is with you know, 1 ml of hydrogen peroxide and 4 mL of water. You know, I can run the reaction again, except now I'll use a more concentrated substrate. So let's say I run it with 2 mL of hydrogen peroxide and only 3 mL water. What am I going to expect from some of your hypotheses? If I increase the substrate concentration, what would probably happen?
Faster reaction.
That
If you have faster reaction
it's
rate.
going to be faster. If it's faster, that means in the same amount of time, we're going to have higher levels of oxygen. We're also going to kind of go longer, and eventually it's going to die as well. Maybe so. This could be maybe 2 mL of H2O2 and 3 mL of water. So this is more concentrated. We have a steeper slope here because it's going faster. The mils of oxygen per second are greater here than the mils of oxygen being produced here. So in fact, that's going to be an important point. Slope is going to be a measure of the reaction rate. The greater the slope, the faster the reaction is going. In both cases, eventually the reactions pooped out. You know, this one had more oxygen by the time it pooped out because there was more hydrogen peroxide to break down in the first place. So we made more oxygen. So we're going to be doing this 5 6 times actually, including the pure water. What we're going to have to do for each of these trials is actually calculate this slope, because that slope is going to be our measure of the reaction rate. Calculating that slope is a little non-trivial. You're going to have to graph things out. You're going to have to do a best fit line. You're going to have to do the rise over run. That's why I don't want to spend all our time today going into the details of actually the mechanics of calculating that slope right now, because I want to make sure we get through all the data collection. But it's going to be pretty, it's just basic math. You go on your graph, you just calculate delta y over delta x. That's all the slope is. Slope is rise over run, change in height over the change in the x-axis. And then what you're going to do after you do this 5 or 6 times, right? One trial, another trial. Again, we need to do a trial at all the different substrate concentrations that we're going to be playing with. Then we're going to have to make another graph. Because what are we really looking for? We're looking at the relationship of what and what.
Reaction rate and the,
With the mass subject part of it.
sorry, the reaction rate of the with the substrate and enzyme.
So what we're really interested in is how substrate concentration affects reaction rate. And that's what we really are looking for in our objective. Each of these graphs of time versus oxygen, those are going to be necessary to get down, but it's only in service of getting these reaction rates, which we can then put on this second graph, which is the one that's actually going to help us answer our question and our objective. So there's going to be 2 steps. The first step is going to be doing these things where you look at time versus oxygen for each of the 6 different trials, which are 6 different concentrations. And then for each of those concentrations, we're going to start from 0 substrate concentration to 100%. Then we have like 20%, 50%, 60%, 80%. But those are just the different ones, trial 1, 2, or trial 6, 1, 2, 3, 4, 5. You'll be mixing up each of these concentrations as you start each trial. It's a little bit simple, so go through it. And then you have to do the more painful process after you get your data, plot it out, get your line, actually on your graph, calculate what is this delta y in terms of moles of oxygen, what is this delta x in time, and then you divide it, it gives you your slope, which is going to be our reaction rate in mils of O2 per second. If there's no substrate, theoretically there's probably no reaction. And let's say we do this and we figure out it's 0.5. And then we calculate this one here. And we say, oh, that was 1. You can see it kind of going up here. So that would be like, oh yeah, look, substrate concentration going up, reaction rates going up. Maybe the next one is even steeper. And now it's up here, and I can see like, whoa, look, continuing my positive correlation. Higher the concentration, higher the reaction rate. What might happen? I know with the thing we talked about. What are there's a couple of possibilities here, Sienna.
The saturation point coverage.
Right, so it might be we don't hit saturation. It might be there's plenty of enzymes for all of our trials, in which case we'll just keep going up until we're done. If we do hit saturation, if we do get to that place where we overwhelm the amount of enzyme that's there, then what happens? A plateau. It means we added even higher concentration, but it's still that same. It didn't go faster. We added even higher. It's still so. Enzyme saturation. Notice that it looks kind of like this. It looks a lot like this, but it's a really different thing. This is x-axis is substrate concentration. Each dot here is a whole experiment that you did, where you graph things out and calculated the slope to get that point. Now, in this reaction here, nothing's dead. The reaction's going. It bubbles like crazy. It's just the bubbles aren't being produced any faster than they were at the previous time. So this is nothing. This is about hitting a maximum reaction rate. So we're not speeding up anymore, but the reaction's still going just fine. And in fact, by the end of the reaction here, we're going to make quite a bit more oxygen at the end of the reaction here, because we'll have had a lot more hydrogen peroxide to have started. Like if I look at this, let's go back to this kind of a graph of mils of oxygen per second. Now what I drew here would look something like this. There's trial 1. And then trial 2, it's steeper. Steeper slope means higher number here, also means it's faster. And ultimately, we made more oxygen as well. Maybe I should color code. Now for this one, it's even faster yet. And we'll also ultimately make more oxygen by the time we finish breaking down all the hydrogen peroxide there. Now, though, this one. It's not going faster. It's going to go longer and ultimately make more oxygen. But the slope is the same because the rate is the same. The mils of oxygen per second being produced is equal. And if I do here again, this one also not going any faster. The slope is the same, but it'll ultimately go for longer and make even more. So when you see same slope, this is an indication that we've hit enzyme saturation. If the slope keeps getting faster with higher concentrations of substrate, then we're not at saturation yet. If you start increasing the concentration, but the slope doesn't get any steeper, the rate isn't getting any faster. But do you also, I want you all to try to see the relationship between. You're gonna have to make these graphs to make this graph. Each of these things, each of these time versus oxygen will give you a plot that you are gonna have to graphically calculate the slope from, which then gives you the numbers you need to make this graph, which is the graph that's actually gonna answer your objective.
And is this something we're doing on Excel?
No, I'd rather you do it by hand.
Really?
Yeah. You'll see why, because there's kind of things that are kind of, it's kind of tricky to do in Excel, because these things are non-linear. They kind of start weird, they start dying. So it's actually easier just to kind of eyeball it and kind of put in your best fit line. I think you are better than the computer at actually figuring out what's the best fit line to figure out the slope best. So, other questions up to this point? You know, this lab is one of the ones that tends to challenge people. I think because of the multiple steps to get to the dependent variable. And our dependent variable is reaction rate. To actually get the reaction rate for each case, you have to make this plot of time versus oxygen, and then do a best fit line and calculate a slope. And then finally, you got the number to put on this graph here. So next class, again, I'll be spending more details, working with you all to make sure you all can do this and feel comfortable. But what we're gonna do today is get our data. We're gonna do the same reaction 6 different times, probably more than that if you mess one up. Not that tricky to mess them up. So you're gonna do this reaction, with 0 substrate, just with distilled water. Do it with 100%, just, pure 3% hydrogen peroxide. And then with all these different dilutions in between. And each case, you're gonna just make a big table full of numbers and record all your data. That data is then going to be where you're gonna start to make these graphs, the time versus oxygen, calculate the slope, ultimately get this and answer the question that you all asked, hopefully. Now I can enzyme saturation. I have enough of these for saturation. Okay, I think trying to get you guys to think about enzyme saturation is easier to actually do it. Let's say I am the enzyme, this is the substrate, and when I get this thing, I break it apart. Right? Let's say I have I can do it like 1 sheet per second. So every sheet. All right. 1 second. 1 second. 1 second. So me alone, I can tear paper 1 sheet per second. Let's say we're all enzymes. And now, if you throw 1 sheet in here, it's still going to get torn in 1 second because it's only 1 person. But let's say I double the concentration. I'm going to put 2 sheets of paper in the room. And here we go. You ready? It's in. Go. Right. We now have double the reaction rate. We're tearing paper 2 sheets per second. I can add in even more. Let's do 4 times. 4 times. So don't go yet. You ready? Go. We now are tearing paper 4 sheets per second. because all of these enzymes are working in parallel and finding a substrate and doing its thing. And I could keep this up until we got up to like, if I added up to 24 sheets in the room, we'd be tearing them 24 sheets per second. But what happens if I start adding in more than 24 sheets? Let's say I added 50 sheets of paper into the room. How fast could we as a class be tearing this paper up? We'd be maxed out at 24 sheets per second. Doesn't matter how much more paper is waiting in the wings. If all us enzymes are working at full capacity, the reaction rate is maxed out. It can't go faster because all the enzymes are doing their thing. Does that make sense? So that's what this saturation is about. It's not about the reaction stopping or even slowing down. It's just though it's going as fast as it can, and even if you start increasing the substrate concentration, the enzymes are going as fast as they can, so they're going to just keep tearing apart the hydrogen peroxide at this maximum rate, depending on the amount of enzyme molecules available, until they finally finish breaking down all the hydrogen peroxide. Hopefully that makes it feel a little more intuitive. So, all right, what we're going to do now is make our